Did you know that one day you might eat a chicken nugget grown entirely in a laboratory rather than raise it in a farm? Traditional livestock farming feeds billions, but it also causes enormous costs, greenhouse gas emissions, deforestation, and animal suffering. So, what if we could grow meat directly from animal cells without raising or slaughtering animals? In this video, we'll explore cultured meat, how it's made, why scientists are excited about it, and the biological processes that make it possible. What is a cultured meat? Cultured meat, also called lab-grown meat or cell-based meat, is real meat produced by growing animal cells outside of the animal body. Scientists start with a tiny sample of animal tissue, isolate muscle stem cells, and then grow these cells in a nutrient-rich environment until they form muscle and fat, the main components of meat. To give the product structure, cells are placed in scaffolds, 3D frameworks that let the cells organize just like they do inside a living animal. So in short, it's not imitation meat, like plant-based burgers. It's real meat, just grown differently. Why do we need it? The motivation behind cultured meat is both ethical and environmental. Livestock farming uses nearly 30% of Earth's land, and 14 to 18 of greenhouse gases come from livestock. Billions of animals are killed each year for food, and as world population rises, meat demand keeps growing. Cultured meat aims to provide the same nutrition and flavor as conventional meat, while reducing environmental impact and animal suffering. Some even call it clean meat. Some even call it a clean meat, a potential solution for sustainable protein production. The process of making a cultured meat consists of 5 main steps, and the step 1 is biopsy. Biopsy is a simple medical procedure where a small piece of tissue is removed for study. In our case, scientists take a tiny sample, usually from muscle from a living animal, often under a local anesthesia. This doesn't hurt an animal. Inside the tissue are muscle stem cells, the key ingredients for growing meat. These cells have an ability to divide many times and develop into different types of cells that make up muscle tissue, just like shown in the video. So what exactly are stem cells? They are undifferentiated cells that can become many other types of cells and can self-renew indefinitely. In animals, there are different levels of potency: totipotent, pluripotent, and multipotent. Totipotent stem cells, the most powerful ones, have the capacity to differentiate into all the different cell types found within the body. Pluripotent stem cells can mature into all cell types, save those in the placenta. And multipotent stem cells can differentiate into several specialized cell types within one lineage. Scientists have developed induced pluripotent stem cells, essentially multipotent blood and skin cells that have been regressed to a pluripotent state, enabling them to differentiate into a greater range of cells. Telomeres, which we have studied in class, the protective caps at the end of chromosomes, also play a key role here. Each time a cell divides, its telomeres get slightly shorter, limiting how many times it can divide. To make cultured meat efficiently, scientists sometimes use younger stem cells or ways to maintain telomere length, ensuring cells can keep dividing long enough to grow substantial tissue. The growth medium. After isolating stem cells, they are placed into a growth medium, a nutrient solution that provides everything they need: sugars for energy, amino acids for proteins, vitamins, minerals, salts, and special growth factors, molecules that tell cells to multiply. Originally, many labs used a serum made from calf blood, FBS, but that defeated the purpose of cruelty-free meat. Today, companies are developing synthetic or plant-based growth media to make production ethical and scalable. Step 4 is growing and structuring the tissue. Cells in the growth medium divide rapidly, forming small clusters. So, we have a lot of cells, but this is not enough to make a meat. They need to organize into the correct structure. That's where scaffolds come in. A scaffold acts like an artificial extracellular matrix, which you also studied in class. The natural 3D network of proteins that support cells in a body. It helps cells attach, align, and sense mechanical signals, which encourage them to differentiate into mature muscle fibers. Scientists sometimes use bioreactors, controlled chambers that supply oxygen, remove waste, and keep the right temperature and pH to grow larger amount of tissue. Once the tissue has developed enough, usually after several weeks, the muscle fibers are harvested. They can be combined with fat cells for better flavor and texture, and then processed just like the regular meat, cooked, seasoned, or shaped into a burger or nuggets. It's real meat at the molecular level. Same proteins, same fats, only without an animal behind it. Where are we now? Cultured meat is still in its early stages. Only a few countries have approved it for sale. That's Singapore, United States, Australia, and Israel. Other countries, including South Korea, are considering approval soon. Meanwhile, some governments like Italy have even moved to restrict or ban it, citing safety and cultural concerns. Costs have fallen dramatically from $330,000 for the first burger in 2013 to under $10 today in pilot labs. Major challenges remain: scaling up production efficiently and minimizing the cost, replacing animal-based growth factors entirely, matching texture and flavor of traditional meat, and consumer acceptance and regulation. Scientists are also exploring genetic and cellular optimization to make the process faster and more sustainable. So, to sum up, in this lesson we've learned that cultured meat is a real meat made by growing animal cells in a lab. The process involves 5 main steps, which you can write your answer in the comments, and if you have any mistakes, I can correct you. We explored biological foundations such as stem cell potency, extracellular matrix, and telomeres, all crucial for understanding how living tissue grows. You can also write in the comments what you understood about these topics. And finally, we explored how cultured meat could reduce environmental damage and animal suffering, but still faces scientific and social challenges. The next time you eat a burger, imagine, could the future of food come not from farms, but from labs? Thank you.